In 1916, a zoologist named Borradaile gave a name to evolution's strangest twist, a world where animals with no relation keep turning into crabs again and again. It has happened at least 5 times, spanning millions of years and rewriting what scientists thought was possible. But if becoming a crab is so inevitable, what invisible force keeps pulling nature back to this same bizarre shape? The answers begin deep in the fossil record with a discovery that shocked the scientific world. In 1916, Lancelot Alexander Borradaile sat in his study, surrounded by preserved crustaceans and anatomical sketches, puzzling over a pattern that defied simple explanation. He noticed that certain crustaceans, separated by millions of years and miles of ocean, kept ending up with the same body plan. A broad, flattened shell, legs arranged for scuttling sideways, and most tellingly, an abdomen curled tightly beneath the body. This tucked-under tail, once long and exposed in their ancestors, now vanished from view, a trait he saw again and again in creatures that should have been distant relatives. Borradaile captured this strange repeating transformation with a single word: carcinization. In his 1916 paper, he described it as the many attempts of nature to evolve a crab, focusing on how elongated, shrimp-like ancestors, especially among the Anomura, a group that includes hermit crabs, had independently evolved into forms nearly indistinguishable from true crabs. What fascinated Borradaile was not just the external similarities. He saw the abdomen, or pleon, reduced and bent beneath the thorax, the carapace widened and flattened, and the walking legs set for rapid, ground-hugging movement. These were not minor tweaks, but sweeping reconfigurations, happening over and over in lineages that had no close connection. The term itself, carcinization, comes from the Greek word for crab, karkinos. Borradaile's definition was precise: a reduction of the abdomen of a macrorhis crustacean, together with a depression and broadening of its cephalothorax. He had no genetic proof, no fossil time machine, only careful observation and a sharp sense for patterns in nature. Yet even without molecular tools, Borradaile recognized that this was more than coincidence. He called attention to king crabs, porcelain crabs, and squat lobsters, all of which had independently arrived at the crab-like form from ancestors that looked nothing like modern crabs. Each time, the abdomen shortened, the shell became a shield, and the animal's shape shifted toward the same end. Borradaile's insight landed at a time when evolutionary biology was still finding its footing. The idea that nature could repeat itself so exactly, sculpting the same design from different raw materials, ran counter to the expectation that evolution was a matter of branching novelty. Yet here was clear evidence that certain solutions were so effective, they kept reappearing. The crab form, with its armored carapace and compact build, seemed to offer a blueprint for survival in the crowded, dangerous world of the seafloor. Borradaile's coinage of carcinization did more than name a curiosity. It reframed how biologists thought about convergence and set off a century-long search for evidence, first in anatomy, then in the fossil record, and eventually in DNA. The word itself became a touchstone for evolutionary puzzles, a reminder that nature's creativity sometimes runs along the same grooves, no matter where the journey begins. In the mountains of central Colombia, a team led by paleontologist Javier Luque uncovered a fossil that would force scientists to rethink everything they thought they knew about the origins of the crab form. The year was 2005. The stone layers of the Paja Formation, pressed and folded by millions of years of geologic change, held the remains of a creature so strange, so unlike anything living today, that Luque named it Callichimera perplexa, the perplexing, beautiful chimera. Dating to around 110 million years ago, deep in the Middle Cretaceous, Callichimera sat in the fossil record as a kind of evolutionary riddle. Its body was barely the size of a thumbnail, yet the details were unmistakable. The carapace was broad and rounded, with paddle-like legs splayed to the side. The eyes were huge, unshielded, and stuck out like headlights. Most striking, the abdomen was already shortened and tucked beneath the body, an unmistakable hallmark of the crab blueprint. But nothing else about Callichimera fit the mold. Its limbs resembled those of swimming crabs, yet the rest of its body carried features seen only in ancient, shrimp-like relatives. This was not a modern crab, nor was it a direct ancestor to any living species. Instead, it was a mosaic, combining traits from distant branches of the crustacean family tree. The discovery of Callichimera did more than add a new species to the fossil catalog. It pushed the timeline of carcinization back by more than 50 million years, revealing that the crab-like form had already emerged in the Cretaceous, long before true crabs began to dominate the world's oceans. The fossil's age, pinned by radiometric dating and ammonite fossils to about 110 million years ago, anchored the pattern of convergence in deep time. Here was proof that nature's crab solution was not a recent twist, but a recurring theme written into the fabric of life itself. Carcinization had roots far deeper than scientists had imagined. For evolutionary biologists, Callichimera became a touchstone. Its blend of features suggested that the path to the crab form was not a straight line, but a network of experiments, some leading to modern crabs, others ending in evolutionary dead ends. The fossil's preservation, with its delicate appendages and fine eye structure, allowed scientists to reconstruct not just its anatomy, but its possible lifestyle. Callichimera likely swam through Cretaceous seas, using its paddle-like limbs for bursts of speed, and its compact body for squeezing into crevices or escaping predators. Javier Luque's find didn't just fill a gap in the fossil record, it raised new questions. If crab-like shapes appeared so early and in such unexpected forms, what drove this repeated transformation? And how many times had nature tried and succeeded at building a crab from scratch? The answers, it turned out, lay not only in the rocks, but in the genetic blueprints of living crustaceans, waiting to be decoded. But before the age of molecular biology, Callichimera stood as a silent witness, proof that the story of carcinization began long before anyone was around to give it a name. In a Los Angeles lab in the early 2000s, Joel Martin and Shane Ahyang faced a problem that had stumped biologists for decades. On the surface, king crabs and hermit crabs seemed worlds apart. King crabs, with their massive, spiny armor and long legs, looked every bit the heavyweight predator. Hermit crabs, by contrast, scuttled about in borrowed shells, soft-bodied and vulnerable. Yet their anatomy, especially the way their abdomens curled and tucked, hinted at a hidden connection. For years, taxonomists grouped king crabs with true crabs, assuming their similarities were a sign of close kinship. The idea that these giants might actually be hermit crabs in disguise seemed almost absurd. Martin and Ahyang decided to go beyond appearances. They turned to molecular phylogenetics, a method that compares DNA sequences across species to reconstruct evolutionary relationships. This approach offered something anatomy alone could not: a genetic family tree built from the inside out. Their team gathered samples from dozens of crab species, including king crabs, hermit crabs, squat lobsters, and the so-called true crabs. They sequenced multiple genes, nuclear and mitochondrial, coding and non-coding, to ensure the results could not be dismissed as a fluke of one stretch of DNA. The data did not just challenge old assumptions, it blew them apart. When the gene sequences were run through multi-locus analyses, the king crabs consistently landed within the hermit crab branch of the tree. Not next to it, not somewhere nearby, right in the middle. In every test, with every gene, the king crab's DNA declared their ancestry. They were highly modified hermit crabs, not close relatives of true crabs at all. The massive, armored king crab had evolved from a soft, shell-dwelling ancestor, an evolutionary leap that required not just a new shell, but a complete overhaul of body structure and lifestyle. This was not a single, lucky result. Over the next few years, as more genes were sequenced and more species added to the analysis, the finding held. The king crab's place among the hermits became one of the best supported results in crustacean phylogenetics. It forced biologists to redraw family trees, rewrite textbooks, and rethink the very rules of how evolution repeats itself. The transformation from hermit to king had happened independently, not as a minor tweak, but as a full-scale reinvention, an event so improbable it could only be explained by powerful selective pressures driving the same outcome from different starting points. Martin and Ahyang's work did more than solve a taxonomic riddle. It provided the first hard, genetic proof that nature's crab-making habit was real, not just a trick of the eye. The crab form had emerged again and again from ancestors that looked nothing like each other, confirming what Borradaile had only suspected. In the world of crustaceans, DNA became the ultimate witness, testifying that the crab blueprint had been drafted and redrafted, each time from scratch. The story of carcinization was no longer a matter of opinion or anatomy. It was a fact, written in the code of life itself. Across the world's oceans, the story of crab making repeats with uncanny precision. Porcelain crabs, for instance, belong to a family that split from squat lobster ancestors sometime in the late Jurassic, more than 140 million years ago. Their bodies flatten and widen, their abdomens curl beneath them, yet they keep the long antennae of their lobster kin, a telltale sign that their crab-like shape is a clever imitation and not a shared inheritance. In Australia, the solitary Lomis hirta, known as the hairy stone crab, stands as a living oddity. Genetic studies place it far from true crabs, yet its body fits the crab blueprint so closely that only a specialist can spot the difference. Coconut crabs, the giants of the Indo-Pacific Islands, take the pattern onto land. Weighing up to 9 pounds, these crustaceans wield massive claws and sport a compact shell, their bodies armored for both climbing palm trees and surviving harsh terrestrial life. Each of these lineages, porcelain, lomus, and coconut, represents a separate evolutionary experiment, arriving at the same body plan through different routes. Counting up the cases, evolutionary biologists now recognize at least 5 independent origins of the crab-like form within decapod crustaceans. The tally includes true crabs called Brachyura, porcelain crabs called Porcellanidae, king crabs called Lithodidae, Lomis hirta, and the coconut crab's extended kin. Some estimates go higher, depending on how strictly scientists define crabness. Even under the most conservative criteria, broad carapace, bent abdomen, fused sternites, the number stands out. Carcinization has happened more often in decapods than most other celebrated examples of convergence. For comparison, the camera-type eye evolved independently about 6 times across all animals. Echolocation, a classic case in mammals, shows up around 60 times, but those events are spread across the entire vertebrate tree. In crustaceans, the crab form has taken over a single order, rewriting the rules for thousands of species. A bar chart of evolutionary convergences places carcinization near the top for morphological extremes. There are 5 or more full transformations, each from ancestors with long, exposed tails and cylindrical bodies, and each ending with a compact, armored crawler built for hugging the seafloor or squeezing into cracks. In some groups, like squat lobsters, the process runs only part way, producing intermediates with flattened bodies but still trailing a visible tail. Elsewhere, the transformation is complete, leaving no hint of the original design except in the genome. The sheer frequency of these events, all within a relatively narrow slice of the animal kingdom, is what turns the crab story from curiosity to mystery. Scientists have cataloged these cases with increasing precision, using both fossil evidence and DNA sequences to confirm that each crab-like lineage started from a different point on the evolutionary map. The odds of so many separate lineages converging on the same solution over and over stretch the imagination. Why this particular shape, and why so often? The pattern suggests that something in the environment, or in the crustacean body plan itself, nudges evolution toward the same destination. The more examples biologists uncover, the more pressing the question becomes. What makes the crab form so irresistible that nature keeps reinventing it? The answer, if there is one, lies not just in the numbers, but in the mechanics of how bodies change and adapt. A tiny crustacean called Borradaile hawaiiensis, barely the length of a grain of rice, holds the key to understanding how evolution can rewrite the body's blueprint. In Nipam Patel's lab, researchers use this translucent amphipod to probe the genetic code that sculpts crustacean form. The focus is on a set of genes known as Hox genes, specifically UBX, ABDA, and ABDB. These genes act like molecular architects, telling each segment of the body what kind of appendage it should grow. In Borradaile, scientists use CRISPR to knock out or tweak these genes, watching as limbs transform before their eyes. When UBX is deactivated, legs that should have become strong, walking appendages instead turn into delicate, feeding limbs. Shift the boundaries of ABDEA, and the middle segments lose their identity, blurring the line between leg and pleopod. ABDB, the gene that usually suppresses leg identity in the abdomen, becomes a master switch. Remove it, and the tail sprouts extra legs, an echo of ancient forms. These experiments reveal how subtle changes in gene regulation can compress the tail, flatten the shell, and rearrange limbs, recapitulating the same suite of traits that define a crab. The genetic toolkit is not unique to crabs. It is present in shrimp, lobsters, and their kin, but the timing and intensity of Hox's gene activity create the dramatic shift from a long-bodied crawler to a compact, armored scuttler. The path from shrimp to crab is not a single leap, but a series of molecular nudges, each one shaping the body closer to that familiar, efficient form. In the lab, the boundaries between crustacean types blur, and the plausibility of repeated crab making becomes clear. The instructions are already in the code, waiting for the right combination to unlock them. Not every crab stays a crab. In the fossil record and in living species, the story sometimes runs in reverse. Frog crabs, for example, belong to the true crab lineage, yet their bodies have stretched out again, their abdomens unfurling and carapaces narrowing, echoing the shape of their distant, long-bodied ancestors. Molecular studies confirm these are not primitive forms, but descendants of fully carcinized crabs that have abandoned the compact design. Similar reversals appear in purse crabs and even within porcelain crab relatives, where the signature tucked abdomen loosens and the body plan drifts back toward a squat lobster shape. These decarcinization events, tracked through DNA and fossils, reveal that the crab form is not an evolutionary endpoint, but a flexible solution, one that can be gained and lost depending on circumstance. This pattern has fueled one of the liveliest debates in evolutionary biology. Some researchers argue that the crab shape is the inevitable outcome of natural selection, the best answer to the challenges of life on the seafloor. Others see it as a statistical mirage, a product of shared developmental pathways and chance, not destiny. The existence of reversals, species that give up their crabness, suggests that what looks like inevitability may really be a question of context, where the best body plan is always shifting. The debate has spilled over from academic journals into popular culture. In 2019, the phrase, "everything becomes a crab," exploded across social media, turning carcinization into a meme. For a moment, the internet made crabs a symbol of evolution's quirks, a shorthand for the strange mix of chance and constraint that shapes life. Yet behind the humor lies a real puzzle. If the crab form keeps reappearing but can also vanish, what does that say about predictability in evolution? Is nature working from a hidden script or just rolling the dice over and over? The evidence leaves the question open, inviting us to look at every sideways scuttle with fresh eyes. Right now, genetic research still uncovers new crab-like forms evolving across the world's oceans. As climate shifts and ecosystems transform, the pressure to adapt only grows. Carcinization reminds us, evolution is not random chaos, but a relentless search for survival's edge. Nature still writes its rules, and sometimes the answer keeps turning out to be a crab. What form do you think the future will favor next?